So we can continue, and the next lecture will be Romualdo Pastor-Satorras, the full professor in applied physics in the Universitat Politècnica de Catalunya, Barcelona, Spain. I don't meet Ronaldo personally, but I remember his paper, Epidemic Processes in Complex Networks, published together with his colleagues, I think 10 years ago, and it was one of the best reviews, if not the unique one on this topic, very relevant at that time and still important for many other applications. Today we will have an opportunity to hear his lecture with the title The Complexity of Moving Animals. But please, Ronaldo, the screen is yours.
Okay, so thank you very much. And first of all, I would like to thank Yuri and the rest of the organizers of this series of lectures for inviting me. It's really a pity that I cannot be in Ukraine, and I look forward very much to just be there in person at some time. Also, maybe it's not a bad thing that it's online because I have a bad cold, and just talking about epidemic spreading, that could be like a bad idea if you meet me right now in person. I'm sorry. So in these lectures, I would like to, let's say, maybe continue with a little bit something similar that Erwin did in his lectures finished a few minutes ago, and it's to explore the complexity of natural and biological systems. Okay, so the idea is that we want to study the complexity of nature, and I mean, it's when we do this, it's maybe appropriate to just remember one important quote is that when you think about the complexity of nature, like plants using quantum mechanics for photosynthesis, a smartphone begins to look like a pretty dumb object. So nature, biological systems are things really very complex, very complicated to study. So here I'll probably go to something much simpler to what we have seen in the previous lectures by Erwin, and I'll just focus in one particular aspect, which is movement of animals. So the idea is that I'll present during this lecture some examples of applications that we have been doing about applying tools of statistical mechanics and complex system science to the study of how animals move and what are the patterns that this movement can take, especially when we consider groups, large groups of animals. So this is like a much simpler specific aspect of nature, just moving animals, and we try to apply these tools. But anyway, even if it's simpler, we must be careful how we apply these tools. And there is also this quote that is relevant for the case, is that for every complex problem, there is an answer that is clear, simple, and wrong. So we must be careful when we apply our tools and to see that we are not making wrong things. Okay, this is a very optimistic table of contents of my lectures. I hope I'll be able to finish it, but if not, I'll stop when the chairman tells me to do so. So I'll go to first a brief introduction of what is this thing of animal movement and why it's interesting. Then I'll explain a little bit about one part of the research that we are doing is that is performing experiments with animals. So I will tell you a little bit about how we do these experiments. Then we'll start with some applications of, let's say, criticality and statistical physics in animal movement. First is like showing that there is a possible phase transition in animal movement when you have like big groups of animals that move together, and what are the effects of social interactions in these transitions. Then we will see a nice application of statistical mechanics, that is the observation of avalanches in moving fish. If I don't have time, which probably I won't, I'll finish this part in my second lecture, and then I'll try to explain you a little bit about how can we model the movement of these groups of animals. So the idea is we are going to study animal movement. So this movement is something that is one of the basic characteristics of living organisms. And they move because they look for food, they look for mates, they have to look for a better place to live when the one they are in it becomes ruined for some reason. They have to move to find, to catch the food. They also have to move to escape predators that want to just eat them. So this movement can take many different forms. Like, for example, we can have, oops, sorry, like this. We can have crawling, we can have running, we can have flying, we can have swimming. Here in particular, I'll be especially interested in this kind of moving, like it's swimming of fish. So animal movement is something that has been like honed by natural selections, and it has been like in every different species adapted for very particular purposes. So we can have some animals that they can fly thousands of miles without resting. And to do so, they had to develop like a way of flying that is energetically with a very low consumption of energy. So they move slowly, consume a little bit of energy, but they can move for very long times. Others have evolved instead like very energetically costly ways of moving. For example, predators that have to catch the prey in a run, maybe 100 kilometers an hour for a few seconds, and the prey that obviously they want to avoid being eaten, and they have developed also speed to evade the predator. So this is something that is very costly energetically. They move very fast, but they can move only for a few seconds. Sorry. So the movement of a single animal is very complex, but the complexity takes more orders of magnitude when we consider social animals, that they move groups, they form groups that move together. This is usually referred in the fields of biology as collective motion. And it's essentially the spontaneous emergence of ordered movement in a system consisting of a large number of moving animals. That, just for simplicity, they can be called like self-propelled agents of particles. Okay? So this is one of these examples. I think Erwin showed a very similar movie. This is a very famous movie called The Starling Murmurations. And this is some city in Sacramento, some village in Sacramento. There are flocks of thousands of starlings, which are small birds that at dusk, they start flying together, forming these fantastic patterns of movement in which there are birds that essentially have very little environmental information. They don't have like long range interactions among them. They don't have telepathy at all. And instead they can just form this organized phase in which they just move together. They can perform like very complex maneuvers, and essentially they never collide. And they look like they were like a single organism with a single brain that was just moving them. And this is calling this aspect of this collective motion is called the flocking of animals. And the idea is this: we have a lot of animals that they move together in a coordinated way, essentially all of them moving in the same average direction. Okay. Now, examples of flocking we can have in birds, in fish, in locusts. They have like also a very famous movie in YouTube and on these nature channels that you might have seen. This the herds of wildebeest that migrate in Africa for thousands of kilometers yearly. We can have also swarms of bacteria that show this kind of flocking behavior, and also we can have also swarms of people. So why flocking? Why animals should be social and move together? Because these have adaptive benefits. For example, it has been argued that when they move together, for them, it is easier to find and capture food. Also, when they move together, and they just easier for them to find, let's say, mates, when you have these flocks of males showing off to attract the females. And also from a more physical point of view, it has been argued that it's aerodynamically favorable for birds to fly together. So they consume less energy when they fly together, forming these special shapes in the air than when they fly alone. In the case of fish, which will be, like say, the main scope and the main subject of my talk. It has been argued that it's also very good from an adaptive point of view for fish to be together and to travel together. Essentially, they can use this flocking aspect of their movement to avoid predators, and they can do it by reducing visibility, by just pretending to be a larger object, and in this case, in this way, they frighten the predator. And also, when they are attacked, they can confuse the predator, offering many moving targets at the same time that maybe some of them will be eaten, but for the majority, if you are in company with a lot of people just running, so your chances of being eaten is smaller than if you are just being chased by your predator. So what is the interest from a physical point of view and a statistical point of view of this collective motion? The thing is that this sort of flocking behavior, you can observe it in a wide range of different kinds of animals, ranging to bacteria to big mammals like wild beasts. These different kinds of animals can have very different sizes, again, from bacteria to large mammals. And you can also have flocks with a very different number of individuals. So you can have flocks with a few tens of birds, or in the case of the workhorse locust swarms, you can have 1 trillion individuals that they are traveling together. So this evidence that there is a similar behavior with these variations of, let's say, space and time scales is something that is reminiscent to the concept of universality in statistical physics and complexity. And this is something that has attracted a lot the attention of statistical physicists and also of applied mathematicians. How do mathematicians and statistical physics study these problems? Essentially analyzing models of moving particles. So we have some simple rules that can affect how you move, how you orient your body when you move, how you interact with your neighbors. When you move, you can also add some random noise, random fluctuations that can account for variations in environmental factors. And there has been like a lot of, let's say, studies and models and a lot of understanding on how collective motion works based in the analysis of these models, which were biologically, physically inspired, but initially mostly theoretical. So what people thought that animals should do when they move. The key point of these models is that these animals are self-propelled particles, so they move. They have some internal energy that they spend in moving, and this places them, as we heard in the last lecture, very far from equilibrium. So we cannot use equilibrium models. We have to take into account that we are looking at things that are far away from equilibrium due to this energy injection. This thing has, let's say, promoted analogies also with the field of active matter. It's a field of condensed matter that deals with the movement, the spontaneous movement of microscopic particles. And there has been like a sort of cross-fertilization between condensed matter physicists and people working in collective motion of animals, in exchange of models and ideas that has been probably beneficial for both fields. So nice thing is that recently the experimental approach has advanced also a lot in parallel with theoretical efforts. And this has been done particular to two recent advances. One is in the field of the technology of position monitoring, especially using GPS. That if you have like a big animal, like a big bird or some big mammal, you can attach the device to this animal, and then you can track his movement just with a resolution of seconds and minutes. This is not possible for the smaller animals, such as fish. So there has been in parallel advances in image acquisition. So we can just, let's say, record movies with very high precision, with very high resolution. And also there has been advancements in the development of softwares that can track these movies and find the individual positions of these moving and marked animals in the movie, either in a natural or an experimental setting. So this has opened the door to, let's say, a new class of data-driven modeling approaches in which you can rely on the information that you get from the real movement of animals instead of just postulating, let's say, reasonable interactions or reasonable forces that you plug then in your model. Okay, so since let's say this is recently a very important issue in this study of animal movement, let's devote some minutes to explain a little bit how this experimental one of these experimental data gathering techniques works, and especially I will introduce you to the experiments with fish. So the idea that experiments with fish are relatively easy to make. You can just work with fish that are not very large, so you don't need like a large facility to experiment with them. In particular, in our lab, we are working with the species black neon tetra, which is like a typical aquarium fish that you can buy for your place. It's rather small, 4 centimeters in body length. This fish likes to form schools of not very large size, so they have this tendency of like, let's say, swimming together. And they also have this tendency, or they also like to swim very close to the surface of the water. So if you combine this with a shallow water tank, you can get conditions in which your fish essentially move in a two-dimensional space, which is much easier to analyze and to model. So this is our experimental setup, which is something relatively cheap. So we have here an experimental tank, it's 1 meter square, with a quarter column of 5 centimeters. We record our films with, let's say, a cheap GoPro camera. And with this camera, we get resolutions of around 3000 times 3000 pixels at 50 frames per second, which is more than reasonable. Then when we have these movies, we have to track them. That is, from the movie, extract the positions of at each frame of all the fish in the tank. This we can do using open source. We do it using an open source software called ID Tracker, which is a very cool piece of software because it relies on machine learning techniques to learn each one of the individuals in the movie. So in this way, by learning the individuals, it's very easy for the software to just distinguish them. And especially resolve cases when two fish, they swim very close together, and in, let's say, not so good software tools, the identities of these two fish, if they travel or they become very close one to the other, the identities can be switched. This is very good, this software, but still it passes a few of these glitches caused by these occlusions. And then there is a little bit of work that must be done by hand to correcting these trajectories. So the most expensive item in this setup is essentially a good computer with a good GPU to run this software. So we can do experiments with these things. And once we have them and we have tracking, we can do some post-processing, like correct for possible deformations due to not perfect orientation of the camera. And then we can compute position, velocity, and accelerations from the positions of the fish. So with this setup, we can do these experiments. The drawback of this experiment is not money. It's not that it's very difficult to take care of this fish. It's not that you must be like a guru in computer science to do that. The problem is, of course, bureaucracy and regulations. So fish are vertebrates, so according to the the European Union, you must fill up a lot of paperwork to do an experiment. And when you have one experiment approved, you cannot do another experiment, so you have to fill another paperwork. So experiments are cheap, are easy, they are quick to do. The problem is the time that you spend with bureaucracy between one experiment and the next. But what can we do? So this is the, like I said, experiments of fish that I will be using. When we have our data, we can look at things. For example, we can look at a single fish moving in our tank. We can look at the velocity and then just confirm one basic thing of biology is that many fish swim using what is called a burst and coast behavior. That is, they just like fish, they swim just performing like a pseudo move of the tail, which just gives them acceleration. And this is called a burst phase. And then they, after this burst, this injection of energy, they slide in the water in what is called the coast or gliding phase. We can just study two fish. What we see is that they try to be close together. So this is a heat map of positions of one fish with respect to another. So they tend to be in front or behind with high probability. And they seem to synchronize their burst and coast swimming in such a way that the peaks of the velocity in one fish seem to coincide more or less with the peaks of the velocity in the other fish. Then from here, we can go on and analyze many fish, but this is something that we will do later. So to start, I would like to present you just an application of, let's say, more theoretical tools of statistical physics to the study of collective motion. And the idea is to explain you one of the first models proposed by Thomas Vicsek in 1995, if I remember correctly, that was used to describe these flocking dynamics in animals by means of a phase transition, an equilibrium phase transition between an ordered and a disordered state. This model is very simple. So we have like animals, fish, whatever, in the simplest case in a continuous two-dimensional space. They have some velocity. The velocity has a fixed modulus. So velocity is defined by the orientation of the angle theta of the velocity. And then what fish do is that they have some radius of attention. So they look at the speed of all the other individuals inside this radius of attention. They compute the average velocity. And they compute the angle of this average velocity. They compute the orientation of the average velocity of the neighbors. So they take, in the next time step, this orientation of the neighbors plus some noise term that can mimic difficulties in estimating the speed of your neighbors or cognitive difficulties in just making these calculations, etc. This is represented by some random noise term with a parameter, tuned with a parameter that gives you the strength of the noise that takes a value between 0 and 1. Takes a value between 0 and 1 because this noise is defined from minus pi to pi, and this is an angle. So adding more than minus pi or pi to the angle, just since it's periodic, it makes no sense. Okay, when you perform simulations of these things, you can look at the order parameter called the polarization, that is defined as the average value of the average modulus, no, the modulus of the average velocity of the flock. This is something that if the fish, the animals, travel in random directions, will be very small, and they all travel in the same directions, will tend to 1. And here, this is a simulation of this model in two-dimensional space. You can see that, let me put from the beginning, they start in a disordered state, but then when they become close together, they start just traveling the same direction in a perfectly ordered flock with a ordered school with a polarization of 1. You can make simulations and compute this order parameter for different values of the noise, and that you see there is some sort of transition between an disordered state, an ordered state, flocking phase for a small noise in which your polarization is large, and a disordered phase for large noise, in which polarization is small, and your animals are traveling in random directions. This happens for some, let's say, threshold, eta c, that depends on particle density and velocity. It's important to notice that in the two-dimensional space, this is not, let's say, a classical second-order phase transition, as it might look, but it instead is something more complex related to first-order phase transitions, in which they can have like what is the name? They can have like solutions, states that they last for some time, and quasi-stable solutions, especially if they are in the form of bands, that they can travel forming bands for some time in the ordered space, and then they just become disordered, and they travel again in bands, etc. So the dynamics of this system in, let's say, in 2 dimensions is more complex than a simple second-order phase transition, as this numerical result might suggest. So this is in the case in which animals just, let's say, they react to, let's say, short ranged interactions, for example, visual cues. so they can only see people around them at a short distance. So all individuals are exactly the same. There are no distinctions between them, and you are only aware of the people that is close to you. But it is this perspective, just a visual short-range cues, just ignores the fact that there are many social animals, and the social ties between animals can make them unequal. For example, a calf might have a very strong tendency to follow her mother, even if she is far away from him. And the calf, can locate the mother, but using sound or a smell or some other cue that has much more long range than visual signals. So if we want to take into account these social interactions, it is better to represent our set of animals in terms of a social network, in which you have like the nodes that stand for the animals, and these edges in the network stand for social interactions. And this sort of animal social network have been measured, for example, by detecting family or feeding interactions between different animals, measuring the time spent together, etc. And this has been observed in different social animals. So in this social network, animal social network, the simplest characteristic is what is called the degree distribution, P of k, which is the probability that one individual is socially connected to k others. In many animal social networks, it has been observed that this degree distribution has long tails. So it decays slower than an exponential or a Gaussian distribution. So in the context of flocking animals, how are these social networks? Well, we can just go to our experiments with fish and measure these social networks. For example, in our set of fish, we can use proximity as a proxy for social contact and just define some, let's say, distance. Let's say, for example, 3 body lengths, and say, okay, each animal is socially connected to all the animals that are close to him by 3 body lengths. And then you can just draw these sort of networks in time. You can collect these networks and just somehow integrate them, and then compute this degree distribution of neighbors, and you see that it's actually long-tailed. This is a fit to a log normal distribution, which is something that decays slower than exponential or Gaussian. And also you can look at a little bit in more detail at the number of contacts between given pairs of animals. If animals were just, let's say, interacting randomly, you expect that your pattern of contacts will be something like this, something completely flat. Instead, what you find in your original data is something like this. There are pairs of fish, like these ones, that have a very large tendency to swim together. And there are others, like these blue ones, that have very small tendency. If you measure, let's say, this distribution of number of contacts between pairs, you get this plot that is, again, also a long-tailed log normal distribution. Can be fitted to a long-tailed log normal distribution. So in order to address this problem, is this social networks important? Okay, what we can do is just take our model, for example, this Vicsek model, and plug it on a social network and see if it has any effect on the phase transition in the social network. So using a log normal distribution is, let's say, mathematically complex. So we will use an approximation to a long tail form, even by a much simpler scale-free or power law degree distribution. So you can just pose your model on your network. You define the network in terms of an adjacency matrix that takes value 1 if nodes I and j are connected, and value 0 otherwise. Since the interactions are given by the network and do not depend on space proximity, so they are decoupled from space. So actually what we can do is just only integrate velocities, not integrate the space. And then this is this Vicsek model, in which now the part that we are averaging are the sum over the nearest neighbors in the topology of the network. So you can do your simulations of this thing, looking at your order parameter. Okay. And C by I, what you see is that when the degree exponent, the exponent of this power law degree distribution is large, essentially these plots of order parameter as a function of noise, essentially they don't change with the size of the network. Instead, when the degree exponent is small, you see that the shape tends to change. Essentially, what it looks like is that this critical point, or this threshold separating order from disorder, seems to move when you increase the size of the system. You can just get more information using a typical trick of critical phenomena is that study the susceptibility. And as you know, in a critical phase transition, the critical point is characterized by a peak in the susceptibility. So if you compute the susceptibility and look at the maximum, you can just define an effective threshold for each value of n. And then you compute the susceptibilities for different values of n, different degree exponents, and what you observe, again, is that when the threshold, the exponent is large, this tends to saturate for large n. When the exponent gamma is small, this effective threshold tends to increase with network size. We can do a little bit better, just applying another tool of statistical mechanics, which is finite size scaling analysis. The idea is that you make a fit of your critical point as a function of network size in this form. So from here, you can compute what is the thermodynamic limit of your critical point. Okay, so in the limit of n very large, this thing will tend to the critical point. You do this thing for different values of gamma, and what you see is that when gamma is larger than 2.5, essentially the threshold is 1, which, remember, is the largest value that the noise parameter can take. And when gamma is smaller than 2.5, you see a threshold in the thermodynamic limit that is smaller than 1. So the idea is that for exponent smaller than 5 over 2, there is a true phase transition. Sorry, when the gamma exponent is larger than 2.5, there is a real thermodynamic phase transition, and there is a critical noise that separates the ordered from the disordered state. Instead, when gamma is smaller than 5 over 2, this maximum noise that you need to disorder your system is 1. So essentially, it's like in the sense of a temperature, if it was an infinite temperature. So in this case, there is no true phase transition in the thermodynamic limit, and the system is always ordered. So here the idea is that these networks, this heterogeneous network with this scale-free power law degree distribution, if the exponent is sufficiently small, they are very good, because they can keep the structure, the ordered state of the system, even if the noise is very, very large. Okay? Now, this is numerics. We would like to do some mathematics. The problem is that the Vicsek model is too complex to solve. It can be more or less solved using macroscopic approximation, using field theories in space, but in networks, it cannot be solved analytically. So just following the philosophy of the statistical physicists, what we can do is just this simplified model of animal motion, simplify even more. And one simplification that we can do is just, okay, this Vicsek model was a vector movement in 2 dimensions. Okay, let's see what happens if I consider movement in 1 dimension for a simple scalar velocity. So this is the essence of the Xerox-Barabashi Vicsek model. So here we have like a Vicsek model, but animals move in 1 dimension. So velocity is a variable that is a scalar. The animals can move, and they update the velocity looking at the average velocity of the networks inside some neighborhood, and then, let's say, rescaling this average velocity using some function. That what this function does is that tries to make the values of the velocity close to -1 or +1. So this is in order to avoid like runaway animals that take infinite velocity and they just go away. So you can have these modulating functions of different forms, and we will consider this one, the sine function. So when the average velocity is 1, is negative, then the animal takes a velocity -1. When the average velocity of neighbors is +1, it takes the value +1. So we can write this model. And another important point, which I was forgotten, is that in this model, we have also a noise term, and now the parameter of the noise term has no limits. Since this is a scalar, now noise can go from 0 to infinity. We can define our model in networks, and make simulations, and we observe a very similar behavior. This is plots of the susceptibility with the peak, and this is plots of the effective threshold in the thermodynamic limit. So the idea is that if gamma is smaller than 2.5, we tend to a threshold that takes a finite value in the thermodynamic limit. Instead, for gamma smaller than 2.5, we have thresholds that diverge with the size of the network. Now, I was promising you math. You can do math. I'm not going to show it because it's a little bit boring. But the idea is that you can compute the threshold analytically and observe analytically within what is called an heterogeneous mean field approximation. And you observe this value. And the funny thing is that this value depends on the moment of the average value of k to the power 3 over 2. Now, if you make this average with a power law degree distribution with exponent gamma, you can see that this average value diverges when gamma is smaller than 5 over 2 and converges to a finite value with gamma is larger than 5 over 2. And this gives us like an explanation, analytical explanation of this behavior that we observe in this model and also that we observe in, let's say, in the Vicsek model on networks. This is a fit with comparing simulations with analytical expression. With this analytical expression, we can see that it works quite well. Now we can do even better. So, we have seen experimentally on fish that not all the pairs of fish have the same number of contacts. So there are fish that they have much more contacts than the average, and there are other fish like this one or this one here that have a much less number of contacts than average. So this number of contacts is something that is not heterogeneous, and this in a network can be represented in terms of a weighted network, in which what we have is that a network and the connections between edges have associated also some weight that gives us the strength of the interaction between this pair of nodes. So we can extend our Vicsek model in the case of weighted networks and, for example, perform simulations on real weighted networks. Here is a set of animal weighted networks, social animal weighted networks. We can perform simulations and compute the thresholds for weighted version of the network, for the binary version of the network. And we observe that in general, in these animal social networks, the presence of weight is bad because it diminishes the threshold. So essentially, you need less noise in the weighted version of the network to destroy the order than in the simple binary version of the network. If we take this CV model, the one-dimensional one, we can also plug it in a weighted network, and then math you can do. And what you obtain, assuming that your weights have this particular form, which is particular, but it's also something very common in many weighted networks observed in the world. So you have the weights that depend on the product of the degrees at the endpoints of the edge to some power alpha. What you get is this solution that has a complex phase diagram in this space parameter gamma alpha. And you can see like the three regions that appear. So we have one region in which the threshold in the thermodynamic limit goes to infinity. And this essentially means that the system is very robust to noise. So you need very large noise to destroy order. We have this other region in which your threshold at the thermodynamic limit is a constant. So you have a clear separation between an ordered and a disordered phase. And also we have this new strange region in which in the thermodynamic limit, the threshold goes to 0. And this means that even a little bit of noise is able to destroy the order in your system. So from a point of view of animals, this region should be very good. This is okay, it can withstand some sort of noise. This is a very bad situation. So animals don't want to be in this region because they will never be able to travel together. Okay? So this is what we can say about this model, Vicsek model of extension. So remember, this is like a very theoretical model in which we just sort of explain the presence of a flocking ordered state as a trade-off between interactions with neighbors and some source of external noise. So let's go to, let's say, another aspect of moving animals is the observation of scale-free avalanches in animal behavior and animal movement. So what does this think of avalanches? So this Jean-Philippe talked a little bit about them in the first lecture. The idea of avalanches is that there are many physical systems that when they are subject to some external drive, they react in an intermittent way. So usually when this small drive starts to operate, they just make like reactions, very small reactions, but from time to time, they just perform a very big reaction. This is the typical behavior of earthquakes. So we have that tectonic stress induces a tectonic movement induces like a stress in the crust of Earth, and that just generates movements that liberate energy. So usually they generate very small movements that liberate a small amount of energy, but sometimes there is a movement that generates a huge amount of energy, and this is one earthquake of a very, very large magnitude. So the main characteristic of this avalanche behavior in earthquakes and also in many other systems in statistical physics is that the distribution of the energy liberated by these avalanches of this earthquake is something that follows a power law. Okay. Can we observe this sort of avalanches in animal behavior? Yes, and these have a particular name in the context of biology, and they are called behavioral cascades. And the idea is that in this group of animals moving together, there is some animal that changes its behavior somehow. And this change of behavior can be transmitted to this neighbor and then transmitted in an avalanche-like way, in such a way that it affects a large number of individuals. And then at some point, it stops. So these behavioral changes can be measured in terms of changes of speed, changes of heading, the direction of movement, and they can be due to response to the presence of a predator, like in this movie, the search of food, but they can also, and this is the thing that we will be studying now here, is that they can also be produced spontaneously in just freely moving animals that have no predators, they are not hungry, whatever. Okay. This was observed, for example, in the case of herds of sheep. So this is an experiment performed by Ginelli and co-authors. And we have like here a bunch of sheep grazing freely. And what they do usually is that they expand, they start walking, they expand, they separate, they disaggregate the group in smaller groups. And then at some point, there is usually one of the sheep that start running like crazy towards the center of the group. And this just triggers an avalanche in which all of them just collapse in a very small group. And then let's say they just become again confident. They start like just growing again. And to the point, another sheep starts another avalanche of condensation. If you look at the surface, the effective surface of the herd, you see that it shows this sort of, let's say, intermittent behavior in the area. This sort of intermittent behavior can also be observed in fish. So if you look at fish, what happens is that they just move more or less okay. But then at some point, there is one fish that just performs a large turn, and this turn can be propagated to the neighbors. And then what you have is like a bunch of fish that they are performing quick turns until they stop, and they again just start swimming, let's say, more or less in a continuous, smooth way. So we can observe these rearrangements, these behavioral shifts in terms of, let's say, the way they turn their speed. So this can be measured like what we call the turning rate, which is essentially the absolute value of the angular velocity of the angular velocity of the heading vector, which we identify as the velocity. Here is the math, and the idea is that you can compute this thing just knowing velocity and acceleration of the fish at each point of time. If you measure this turning rate, it has some distribution that has this form. So it has like for small values, it's something that is essentially constant, and these very small values of turns essentially means a smooth trajectory. Okay, but there is some threshold around 0.1, 0.5 in which this decreases. And then what you have is like large turns. So the idea is that one can identify or try to identify these large turns as turns larger than some threshold, which are infrequent turns that can lead to reorientations, sudden reorientations of the movement of the school, while you disregard these small values that are essentially compatible with smooth trajectories. So if you, for example, measure this number of fish that turn at each frame in our recordings, perform a turn larger than some threshold, you see that effectively this has this intermittent pattern, which is typical of, let's say, avalanche dynamics. Here nothing happens, and then a small event, and then a large event, and then here another large event. So something that resembles the plot that I show you for earthquakes. So I have until 15:50 now. So it's 5 more minutes.
Right.
5 more minutes. Okay. Oh, all right. So when we have this idea, we can just define what are turning avalanches. And the idea, okay, let's fix a threshold of the turning rate, and then just look at the frames or movie. And for each frame, we measure the number of fish that perform a turn larger than this threshold. So for example, here, no one. Here, fish 14 perform a large turn, fish 13, fish 7 and 33, etc. So when we have this thing, we can just identify an avalanche, like a stretch of frames, consecutive frames, in which we have fish performing a large turn. And this stretch is separated by 2 frames in which no fish perform a large turn. So we define the duration of the avalanche as the number of frames involved, and the size as the total number of fish. For example, in this case, is 1, 2, 3, 4, 5, 6, size 7. Okay. And also we can define an inter-event time, which is the time between the end of an avalanche and the start of the following one. Okay, what we should expect. So imagine that we know nothing, we assume there are no correlations. We can consider that the fish are performing turns independently and in an uncorrelated way in time. So every time that a fish performs a turn, it just draws at random some turning rate from this distribution. So from here, we can just construct a null model of avalanche evolution and compute the distribution of durations, the distribution of inter-event times, distribution of sizes, and the important thing is that all of these distributions have an exponential form, which is essentially what we expect from something which is uncorrelated and probably can be associated to a simple Poisson process. When we do our experiments instead, we compute our avalanches, what we observe is there are no exponential distributions. Actually, these distributions have a power law form, which is what you observe in avalanche systems with avalanche dynamics in nature. With some exponents that, well, for the distribution of sizes, you have an exponent around 2. For the distribution of times, you have an exponent between 2.5 and 3. These exponents change a little bit, but not too much, depending on the threshold that you impose to compute the avalanches. Okay. Okay, these power law forms, okay, I must be honest, this is not the longest power law I have ever seen in nature. There are others, other nature systems in nature in which you have 4, 5, 6 orders of magnitude in a power law representation. Here we have not. Essentially because these are experiments made with at most 50 fish. We have strong finite size effects. So we can do different things to check if this description in terms of power laws is correct. So one first thing we can do is to look at the average size of avalanches of a fixed duration t. If you do some math, you expect that this average size will grow as a power law with time, with an exponent which is related with the exponents of the size and time distributions. You make this calculation and you see that indeed they follow a power law with an empirical exponent that is very similar to the one that you can compute using this theoretical prediction. Another check that we can do is to just evidence the scale-free nature of these avalanches is to use what is called the Sedna scaling. The idea of the Sedna scaling is that you look at the shape of an avalanche, of the avalanche, and small t of capital T, that is the number of active individuals at frame t during the evolution of an avalanche of duration capital T, for all these values t from 1 to t. If one assume a scaling form for this shape, doing some mathematical calculations, you can see that this scaling form must have an exponent that is equal to this exponent here, m, like this, minus 1. Okay, if you do your things and you check this scaling, you see that within our experimental error bars, you see that these shapes actually fulfill this Sedna collapse. Of course, you must use here, let's say, avalanches that are within the scale-free region. So we can see that here we have like a scale-free form at the beginning. Here we have some bump. So to observe our Sedna scaling, we must use avalanches that are within this power law regime. But okay, I have this power law regime. What is the meaning of this bump that I have over here? Okay, to explain this bump, what one can do is just to look at the effects of walls. The idea is that we can look at the density of avalanches as a function of the position of the center of mass of the avalanche with respect to the walls. And you can see that there are more avalanches triggered in the bulk than in the walls. But the avalanches that are triggered close to the walls tend to have a much larger size. So these very large avalanches might be the ones that just break our power law behavior. And if we do just an analysis of avalanches in the middle of the tank, just regarding the ones that started close to the walls, you observe a much better power law behavior, in which this bump here, this shoulder, is, let's say, just diminished a lot. Okay, I think that's it. And maybe we stop here, 10 minutes. Judith or Mr. Chairman?
Yeah, it's okay. I think it's a good idea to make the break. But if someone would like to ask some question, please, few questions. Oh, I see Yuri, please.
Thank you. You mentioned avalanches and the exponents. And
Yes.
my question is, if you look for different species, say fish, insects, mammals, are these exponents universal for different species too or not?
I cannot tell you because we only have one species.
I see.
If somebody makes experiments or another school, but what I'm sure is that you must consider schooling fishes. If you take fishes that don't form schools, they probably will not have this tendency of move in a correlated way or react to this behavioral shift. So you need another species that is also flocking. But I mean, it's already a lot of work and a lot of difficulty to perform experiments with one single species. Maybe in some years when all the specimens that we have are died of old age or they are going into pension, we can just think about making experiments with a different species. But so far, I have no idea. It's a good question, but I have no idea.
Thank you.
So we will continue in 6 minutes.